Instrumentation used in flame photometry. The flame spectrophotometric procedure requires the following essential apparatus. For flame emission spectroscopy, a nebulizer burner system is required, which produces gaseous metal atoms by using a suitable combustion flame. This flame involves a fuel gas and oxidant mixture. However, with non-flame cells, burners is not required. Second important requirement for flame photometry is a spectrophotometer system, which includes a suitable optical system, a photosensitive detector, and a display device for the output from the detector. Modulated resonance line source is also required. The instrument which is to be used in flame photometry, it very much resembles the atomic absorption spectrometer. A typical flame photometer is shown in the figure below. It is composed of a pressure regulator and flow meter for fuel gases, an atomizer, a burner, mirror, slit, optical system, photosensitive detector, and recording output. Uh, we will see each and every part of the flame photometer one by one. Pressure regulator and flow meter. These are used for the proper adjustment of pressure and flow of gases. A 10-pound gauze for fuel and 25-pound gauze for oxygen are needed. Double diaphragm and needle valves are used to control pressure. To control the gas flow, a rotameter is also inserted in the gas line. A flow rate of 2 to 10 feet per hour is used for majority of times. Atomizer. The atomizer is used to introduce a liquid sample into the flame at stable and reproducible rates. Atomizers are classified as those which introduce the spray into a condensing chamber for removing large droplets, and those which introduce the spray directly into the flame. About 4 to 25 ml of sample per minute is required, of which 5% reaches the flame. Another method involves a slurry of powder material in isopropanol for spraying. Burners. The flame used in flame photometer must possess the following functions. The flame should possess the ability to evaporate the liquid droplets from the sample solution, resulting in the formation of solid residue. The flame should decompose the solid into atoms, and the flame must have the capability to excite the atoms and cause them to emit radiant energy. Flame temperature is primarily responsible for the occurrence of all these above processes, and this flame temperature is controlled by the type of fuel and oxidant, fuel to oxidant ratio, type of the solvent used, amount of the solvent which enters the flame, type of the burner employed in flame photometer, and the particular region in flame which is to be focused into the entrance slit of the spectral isolation units. Mirrors. The radiation from the flame is emitted in all directions in space. A major portion of emitted radiation therefore does not reach the detectors. In order to increase the amount of radiation reaching the detector, a concave mirror is set behind the burner. The focal point of mirror lies at the entrance of the monochromator. Next, which is used in flame photometer, are the slits. The entrance slit is kept between the flame and optical system. This slit permits the radiation from the flame and the reflection of the flame to enter the optical system. The exit slit is kept between the monochromator and the detector. Exit slit allows only a selected wavelength range from the radiation traveling from the monochromator to the detector. It also prevents the entry of interfering lines. Next, Part which is used is the optical system. The optical system functions as a collector and monochromator of light and focuses it on a photosensitive detector. The light is focused on the entire detector by a concave mirror by adjustment of the flame. Use of absorption or interference filters isolates the characteristic radiations used. Better isolation is achieved by using a monochromator. A good slit with a narrow opening is necessary. Photosensitive detectors. Photosensitive detectors are another part of the instrumentation in flame photometry. These detectors, such as barrier layer cell, are not useful because the response is not amplifiable. These detectors produce an electrical signal from the radiation falling on them. Flame photometers restricts the bandwidth of the radiant energy reaching the detectors, so have to employ phototubes and amplifier units to boost the output. Photoelectron multiplier tubes are the best to use. The readout systems available include meters, charts, recorders, and digital displays. The instruments available are both of single beam and double beam type. Single beam flame spectrometers contain only one set of optics. The light emitted from the flame is collected by a reflector and focused by a lens through optical filters into a single photo detector. Or the light from the burner passes into a monochromator and radiation leaving the exit slit is focused onto the detectors. In the double beam flame spectrometers, uh, a second light path is employed for the light emitted by the internal standard element, which is added to each test solution and calibration standard. The signal of one detector opposes that of the other through a suitable indicating device to provide a ratio method of comparing the light intensities between the internal standard and analysis elements. The sample solution is sucked by an atomizer operated by one of the flame producing gases and fed into the flame. The emitted light is collected by a parabolic mirror and a lens. It is then passed through a monochromator or filter. The light of the selected wavelength strikes a photodetector and the magnitude of the electrical signal developed is read on a meter. The flow rates and the ratio of fuel to oxidant are adjusted by introducing pressure regulators and flow meters in the gas line. The spectral lines corresponding to low excitation energies are produced. The flame spectrum so formed is very simple.